Today, we're going to discuss the process of glycolysis, the link reaction, and how they relate to an aspect of theory of knowledge. First, we're going to be covering glycolysis. Glycolysis takes place in the cytoplasm of the cell, and its general outline is the oxidation of glucose to 2 pyruvates with net production of 2 ATPs and 2 NADH. The first stage is the investment phase, where 2 ATPs attach to a 6-carbon glucose molecule, phosphorylating hexose to hexose biphosphate. A phosphate group is removed from ATP to provide the energy for phosphorylation. The phosphate groups allow for a stronger interaction between the hexose and its enzyme in later reactions. After the hexose biphosphate is formed, it undergoes lysis and it is broken down into 2 3-carbon molecules, each attached to a phosphate group. This intermediate is called phosphoglyceraldehyde, and it is also called triose phosphate. The phosphate group allows the sugar to form stronger interactions with the next enzyme down the pathway. Now we enter the payoff phase. This is the main oxidative stage of glycolysis. In a coupled reaction, hydrogen groups are removed from the triose phosphate to reduce an NAD+ molecule into NADH. The resulting energy released by the triose phosphate is used to phosphorylate 2 ATPs. Notice that 1 NADH and 2 ATPs are produced by each triose phosphate, so the net outcome is 2 NADH and 4 ATPs by 2 triose phosphate. The triose phosphates then become pyruvate. So overall, the reactant of 1 glucose molecule, 2 NAD+, 2 ATP, yields the products of 2 pyruvate, 2 NADH, and 2 ATPs. Intermediates involved are hexose, hexose biphosphate, and triose phosphate. No enzymes or oxygens are required. During the process, although 2 ATPs are used, the production of 4 ATPs eventually results in a net gain of 2 ATPs.
After glycolysis is completed, in order to move to the Krebs cycle, a link reaction must occur, and it is the first stage in aerobic respiration. First, pyruvate is transported into the matrix of the mitochondria from the cytoplasm via transport protein. After the pyruvate enters the mitochondrial matrix, it is decarboxylated by the enzyme pyruvate decarboxylase, and a single carbon is released. A water molecule is broken down into 2 hydrogen and 1 oxygen, where the oxygen combines with the 1 carbon from pyruvate decarboxylation to form carbon dioxide. The hydrogen atoms then reduce the NAD+ molecule to NADH plus H+. The remaining 2-carbon compound, also called the acetyl, then combines with the large coenzyme A to form acetyl-CoA. This product then enters the Krebs cycle, which is the next stage in aerobic respiration. So overall, the reactant of 2 pyruvates and 2 coenzyme A's yields 2 acetyl-CoAs, 2 carbon dioxides, and 2 NADH plus H+.
Now we're going to bring in an aspect in theory of knowledge. Glycolysis has many real-life applications in the areas of the natural sciences, particularly in the field of medical diagnostics. Malignant tumor cells perform glycolysis at a rate that is 10 times faster than the non-cancerous cells. Since cancer cells or tumors have limited oxygen supplies due to the capillaries, these cells rely on anaerobic metabolic processes such as glycolysis for ATP. Some tumor cells overexpress specific glycolytic enzymes that results in higher rates of glycolysis. These enzymes, unlike traditional enzymes, often lack certain feedback inhibition mechanisms. The increase in glycolytic activity ultimately counteracts the effects of hypoxia, which is the lack of oxygen, by generating sufficient ATP from this anaerobic pathway. This is discovered by Otto Warburg in the 1930s. He called it the Warburg hypothesis. The high rate of glycolysis can be a sign that is useful in diagnosing cancer. By injecting radioactive deoxyglucose and taking a PET scan, we can visualize the location of cancer. This method essentially serves as a way of knowing where we improve our perception of the cancer. Knowledge of glycolysis and the link reaction therefore helps researchers figure out ways to cure and contain cancer. Adopting the ketogenic diet, which is high in fat, high in protein, and low in carbohydrates, is one of the new proposals in an attempt to starve out the cancer cells, which relies on glycolysis for energy. That concludes our presentation on glycolysis and the link reaction. Thank you for watching.